Assalamu alaikum and hi. My name is Muhammad Afif bin Azharin and I am from RAS 2014A. I will explain about the introduction to the Western blotting and sample preparation to the Western blotting. First of all, the transfer of a macromolecule such as acid and protein to solid phase membranous support is termed blotting. Protein resolved by sodium dodecyl sulfate polyacrylamide gel electrophoresis, which is SDS pitch, are transferred to membrane composed of either nitrocellulose or a polyvinylidene difluoride (PVDF) using electric current, also referred to as a protein blotting or immunoblotting. Western blotting is a powerful method for studying protein in a sample of a tissue, in a sample of cell or a tissue extract. Next, I will explain about the sample preparation in a general for Western blotting. Preparing the sample includes protein extraction from cell or a tissue and estimation of a protein concentration. Various lysis buffer containing detergent and protease and phosphate inhibitors may be used to solubilize protein from whole tissue or a tissue culture extract. The total protein in the sample may be quantified spectrophotometrically at 595 nanometer using a Bradford reagent.
Before the analytical technique Western blot comes into play, everything starts with an SDS page. Upon treatment with SDS, all proteins in a sample are denatured and covered by negative charge. The proteins have a similar master-charge ratio and travel through the gel to the positively charged anode. This allows to separate proteins according to their molecular weight. After staining the polyacrylamide gel, protein bands become visible. Marker proteins as a reference help to determine the molecular weight of proteins in the sample. This band here, for example, contains proteins with a molecular weight of approximately 60 kilodalton. However, they will not reveal which proteins are present in this sample. It could be the protein of interest which has a molecular weight in this range, but the band could have also been caused by other proteins. To confirm the presence of this specific protein, a Western blot has to be performed. The detection of the protein of interest is based on antibodies designed to exclusively bind epitopes of the target protein. The Western blot setup starts with the gel containing the separated proteins. This is placed on top of a membrane, often a nitrocellulose membrane. This membrane is capable to bind large amounts of protein. Later, the idea is to transfer the proteins from the inside of the gel into the membrane. This gel membrane construct is flanked on both sides by filter papers. All parts here are soaked in buffer to facilitate the electricity transmission later. Finally, everything is fixed between the cathode and anode. The electric current spools the negatively charged proteins which are located in the gel to the positively charged anode. This transfers the proteins onto the membrane.
From now on, the membrane has to be incubated several times. The first step includes blocking. Therefore, the membrane is incubated with milk or BSA. Blocking should prevent antibodies from unspecifically binding to the membrane or to other proteins. After blocking, the membrane can be incubated with the primary antibody. This antibody should be selected to only bind to the protein of interest. After incubation with this primary antibody, a washing step will wash away the unbound antibodies while not removing the antibodies that are bound to the protein of interest. Next step involves the incubation with a secondary antibody which has to be specific to the primary antibody. After washing, only those secondary antibodies remain which are bound to the primary antibody. The detection of the protein of interest is the last step in Western blot analysis. For that purpose, the secondary antibodies are conjugated to an enzyme. For many Western blot assays, secondary antibodies are conjugated with horseradish peroxidase. The protein of interest is bound by a primary antibody. The secondary antibody binds to the primary antibody, and when the membrane is incubated in a substrate mixture, the enzyme converts the substrate, thereby producing a semi-luminescent signal that visualizes the protein band. The presence of the target protein in these samples has been confirmed.
Assalamualaikum, my name is Muhammad Izanaim bin Mat Yazid. In this part, I'm going to talk about the application of the Western blotting. Western blotting is a widely used technique in the molecular biology for the detection and analysis of specific protein in a sample. It has a wide range of application in the field of biology and biomedicine. Here are the most common applications of the Western blotting. Firstly, identification of specific protein. Western blotting is used to identify a specific protein of the interest in a complex mixture of protein. This can be done by using a specific antibodies that bind to the protein of interest. The protein is then visualized on the gel through the fluorescent detection method. Second is the quantification of protein expression. Western blotting can be used to determine the amount of specific protein in a sample, which can be used to study change in the protein expression under the different condition or in the different cell types. It is particularly used for the studying change in the protein expression in response to a specific treatment, disease condition, or genetic modification. Next is the analysis of protein localization. Western blotting can be used to analyze the subcellular localization of protein by using a specific antibodies that recognize the protein of interest and by using subcellular fractionation technique to separate the organelles or subcellular compartments. Fourth is the confirmatory testing of diagnosis test. Western blotting is used in confirmatory testing of diagnosis tests such as HIV testing, Lyme disease, and West Nile virus. Western blotting is a confirmatory test that can use in confirm to the result of the other tests such as the ELISA that may have a higher rate of false positive. Lastly is the analysis of antibody specificity. Western blotting can be used to analyze the specificity of antibody and to confirm that the antibody is specific for the protein of interest. This can be done by using the Western blotting to detect the protein of interest in different samples and using in a different method. In conclusion, Western blotting is a powerful and widely used technique in molecular biology for the detection and analysis of specific protein in a sample. It is a versatile technique that can be used for a variety of applications. One of the advantages of Western blotting is it has a high sensitivity which allows for the detection of low level of protein in a sample. It also allows for the visualization of protein in their native conformation, which can provide important information about protein function and regulation. However, Western blotting also has some limitation. It can be a time-consuming and labor-intensive, and it can be difficult to accurately quantify the protein expression level. Additionally, it is limited by the specificity and sensitivity of the antibodies used for the detection, and it can be difficult to unambiguously identify the protein based on their migration pattern on a gel. Overall, Western blotting is a valuable technique that has played a key role in many discoveries in molecular biology and has a wide range of application in the field of biology and biomedicine. Thank you.